Hi dear students, hope all of you are fine. So let's continue learning biology. Today we are heading to another topic from molecular basis of inheritance. Last classes I think the topics are clear for you and DNA packaging also clear, and now you know some molecular level concepts of inheritance. You know our chromosomes are made up of what? DNA and some amount of protein, histone, non-histone proteins. So in the last classes you have learned Mendelian genetics. Mendel has first found factors are controlling our characters, and later Sutton and Boveri confirmed that chromosomes and factors show some parallelism in characters. So they have supported that these factors might be located in chromosomes. Okay. Then further, beyond the concept, you have learned that the structure of chromosomes consists of, it's made up of DNA and certain proteins. And anyway, you must know who has started the work in searching genetic material. Okay, how it made clear that DNA is our genetic material, not protein, not RNA, not any other biomolecules. In our cell, there are number of biomolecules. Among this, while DNA only controls our genetic characters. Now you know gene is made up of DNA. Okay. Then what made this discovery DNA as the genetic material? DNA was first observed by or isolated by whom? Frederick Miescher. You have studied in a lower class. And he called it as nucleic acid. I mean, since it's acidic in nature, nucleic acid, you remember, right? Anyway, here we discuss the search for genetic material. Okay, the notable attempt is given by Frederick Griffith. Okay, so you can learn it under Griffith's experiment. It's the most popular experiment on genetics, Griffith's transformation experiment. Okay, so let us discuss Griffith's experiment first. Okay, and after that we will discuss one more experiment that clearly proved DNA is the genetic material. Okay, first Frederick Griffith's experiment. He has conducted the experiment in 1928. Okay, he conducted experiments using Streptococcus pneumoniae bacteria. Okay, for his experiment he used the bacterium Streptococcus pneumoniae. Okay, and he used mice for his experiment. See, Griffith grew this bacteria in culture plate in his laboratory. And he has found that this bacteria, Streptococcus pneumoniae, are producing two types of colonies. Okay, there are two different strains or variants for the same bacterium. The same bacterium can form two colonies or two variants, two types of forms. One form he has found it is shiny. Okay, one of these two forms, two types were found very shiny in the culture plate. Okay, it produced shiny colonies because they had a polysaccharide coat around it. A shiny coat, mucus or polysaccharide coat was there for this particular bacterium. So it remained very shiny, smooth, and Griffith named it as S strain. Okay, and the other type of bacterium in his culture plate, he named it as R strain. What may be the reason? It is rough. Okay, it is not having a smooth, shiny polysaccharide coat. So this is having without the coat, and this is the rough one, not the shiny or smooth one, and he named it as R strain. So Griffith found two types of strains for this bacterium, S strain and R strain. Okay, so then what is more interesting is he found that the S strain only is virulent, R strain is non-virulent. That means only the S strain is causing the disease pneumonia. Okay, this is the only virulent strain. Okay, the other one, R strain, was found non-virulent. Okay, then come to this experiment, Griffith's experiment. Okay, here I'm just removing this. Keep in mind, S strain had a polysaccharide caught around it, and that is found virulent, causing the disease. R strain without the cord was found non-virulent. Okay, and in the Griffith's experiment, in the first step, first set of experiment, he has used the S strain and injected S strain bacterium into mice. Okay, he has injected it into mice. What will happen? S strain is virulent, so it caused disease. So what happened? Mice died. Okay, then R strain, he has injected R strain into mice. Okay, it is non-virulent. What will happen? Mice lived. Mice lived. And in the second experiment, second set of experiment, he killed the S strain by heating. Okay, in the second experiment, he used heat-killed S strain and injected it into mice. Heat-killed S strain. Okay, what will happen? By heating, that property is lost. Okay, the mice lived. Okay, so the result, mice live. And in the third set of experiment, he has used heat-killed S strain along with R strain, mix it. Okay, plus R strain was used. Okay, what do you expect? Injected into mice. What's our expectation? We expect the mice live. But what happened? From when the mixed form it is used, it happened, the mice died. Okay, and this is what the different steps of Griffith's experiment. The steps are very clear. I think I will not repeat it. And from this experiment, the last step you have to, last set of experiment you have to notice well. And based on this set, Griffith has introduced the concept of transforming principle. Griffith is most popular with this particular concept, that is transforming principle. Keep in mind and understand what is transforming principle. Okay? Then listen here. When heat-killed S strain is used along with R strain, according to Griffith, something from the heat-killed S strain transferred to R strain, and this made R strain to develop a polysaccharide cord and become virulent. Okay, and he called that something as transforming principle. So according to Griffith, the transforming principle from the heat-killed S strain transformed the R strain to S strain. The R strain became S strain so that it caused the disease. Okay, this is what is Griffith's idea he conveyed to us, and these are the different steps of his experiment. But actually, we are not getting any idea whether it's DNA, RNA, protein, or which is the genetic material. This is the starting book. Later, 3 other scientists worked on this and they formulated the next concept. That's what we are going to discuss. I think Griffith's experiment is clear. Griffith's experiment is related to transformation of R strain by heat killed S strain. Heat killing has spoiled that property, violent nature, but when it is used along with the R strain, it converted R strain to S strain. Again became violent. Okay. Then how the other 3 scientists confirmed it. The transforming principle they have studied further. Okay. These scientists are Avery, MacLeod and McCarthy. They did the experiment in 1933, 44, that period, between that period. So, Oswald Avery, Colin MacLeod and Macklin McCarthy in 1933, they have worked further on Griffith's transforming principle. Okay, in order to get the biochemical nature of that. What is the transforming principle? To get that, they worked on Griffith's experimental transforming principle. Okay, so in the first experiment, they have conducted 2 sets of experiment. Okay, in the first set of experiment, Avery, MacLeod and McCarthy, okay, they purified Griffith's heat killed S strain. Okay, they purified the heat killed S strain, and from this they have isolated all the biomolecules, proteins, lipids, okay, RNA by centrifugation. They have isolated these biomolecules from this. So, these 3 scientists first purified heat killed S strain of Griffith and they separated proteins, lipids, RNA, DNA like all biomolecules in that. And then from this heat killed S strain they have isolated this. Then this protein is injected to R strain, lipid is injected to R strain, RNA is injected to R strain, no change for R strain. R strain remained as R strain, no any conversion. But when DNA from this S strain injected to R strain became what? S strain. Transformation happened. Is it clear? So what happened? When they purified the biochemicals and separately injected biochemicals from S strain and separately injected into mice, there was no change, no conversion. I mean not mice, separately injected into R strain. There was no transformation for R strain for lipid, protein, RNA. But when DNA from heat killed S strain is injected to R strain has transformed into S strain. So, what is changing R strain to S strain? It is DNA from the heat killed S strain. So, genetic material is DNA is first proved by like this experiment. Okay. So, in order to confirm this experiment, they have conducted a second experiment. Is the first experiment clear? Heat killed S strain is purified and all the biochemicals were isolated. Okay, and separately injected into R strain. But when R strain is injected with the DNA from this, then only that viral nature is regained, became S strain. Okay, so DNA is the genetic material, they proved. The transforming principle is DNA, they proved. And in order to confirm that, the second experiment, they have digested all this, S strain biochemicals, they isolated, S strain, to the S strain, heat killed S strain they have taken to a test tube. Okay, heat killed S strain. And when this is digested with lipase, okay, lipase enzyme is added means all the lipid inside this will be digested. Then when it is injected to R strain, it did not affect the change. I mean, when lipid is digested with lipase, when protein is digested with protease, when carbohydrate is digested with carbohydrates, when RNA is digested with RNAs, okay, RNA is digested with RNAs, what happened? This all biomolecules will be digested inside this. There won't be no lipase, no protease, no carbohydrates, no RNA. But it did not affect the transformation. Still, it is transformed. When this is injected, R strain is changing to S strain. But when DNase is used, what is DNase? DNase is a DNA digesting enzyme. When DNase is used, what happened? All the DNA inside it is digested. No more DNA. So, when this is injected to R strain, S strain is not formed. That means that when DNA is digested with DNase enzyme, no transformation became possible. That means DNA is responsible for the transformation, not lipid, not protein, not carbohydrate, not RNA. Okay? With these two experiments, they have proved that DNA is the transforming principle which transformed R strain to S strain, not any other biomolecules. So, you have to study thoroughly Griffith's transforming principle experiment, transformation experiments, and how these three other scientists proved or biochemically characterized it. Which is the biochemical, which is that chemical. They proved it is DNA. Okay, this is the first experiment. Okay, this is the confirmation experiment. Two experiments you have to learn. So actually this scientists, three scientists confirmed DNA as the genetic material. Okay, we have a confirmation experiment, the experiment which unequivocally proved DNA as genetic material, that is by Hershey and Chase. Okay, Alfred Hershey and Martha Chase. Before moving to that experiment, you know there are mainly DNA is the main genetic material, and also in some organisms RNA is the genetic material. And DNA versus RNA, that is a small concept. We will just finish it and then move to the next topic. Then when we compare DNA and RNA as the genetic material, which one is the best? That is what we are moving to. Okay, DNA is genetic material in most of the organisms, whereas RNA is genetic material in some organisms. Can you name some of the organisms in which RNA is the genetic material? You have learned retroviruses. Yeah, AIDS virus, HIV, the recent coronavirus. Then, so many bacteriophages also. QB bacteriophage. Okay, tobacco mosaic virus. Like so, there are organisms, especially viruses, which have RNA as genetic material. Anyway, DNA is the major genetic material. What makes DNA more superior to RNA as a genetic material? That is what our question is. Before that, what are the features of a genetic material? We have to just see the characteristics of a genetic material, then how DNA and RNA fit for that. Okay, genetic material characteristics. When we discuss, the first and most important thing, a genetic material must be able to produce its replica. Okay, when it's moving from one generation to next generation, it should be able to produce its copy. So, a genetic material must be able to undergo replication and produce the replica. You know, both DNA and RNA can replicate. So, DNA and RNA both can be used as genetic material when we consider the first feature. Okay, the first important characteristic, whether it's a genetic material, it should be able to produce its replica, its copy, by the process of replication. Both the DNA and RNA can replicate, so therefore both are fit for the first feature. Second characteristic, if this is a genetic material, it should be chemically and structurally stable. Okay? A genetic material must be stable chemically and structurally. When we discuss this feature, DNA has a great advantage over RNA. DNA is stable compared to RNA. You know, in RNA, the sugar is ribose sugar. In the second carbon, there is a hydroxyl group in RNA, the ribose sugar. Whereas in deoxyribose sugar, this hydroxyl group is not there. There is hydrogen only. The presence of second prime hydroxyl group, second carbon hydroxyl group, makes RNA more reactive. Okay. Whereas DNA is not having that hydroxyl group and DNA is less reactive. So DNA is more stable, the best genetic material in this regard compared to RNA. Okay. Then the next feature, if it's a genetic material, it should undergo slow changes and should provide scope for evolution. Okay. It should provide some scope for slow mutation, not a large mutation. And when we consider this feature, which is giving slow mutation and scope for evolution is DNA. Being reactive, RNA may undergo rapid changes. So this is not best fit for giving evolution. Okay, it can give evolution, it not close there, but it will be a sudden change. You know the coronavirus. When it has started from China, it had one variant. But now you know there are different variants reported. It is very suddenly mutated and is getting converted to other variants. Okay. Likewise, if it's RNA genetic material, it can undergo somewhat speedy mutation and changes. A genetic material should should give chances for slow evolution. Okay. Gradually, we need a variation. And another important concern, the fourth characteristic, it should be able to express. What is the ultimate aim of a genetic material? What is the ultimate aim of a genetic material? Okay, what our genetic material give or express our characters. A gene is responsible for skin color, eye characteristic, hair color, everything. So it should be able to express in the form of a character. Okay, and both this DNA and RNA are able to express by producing protein. Okay, the RNA can directly form protein, DNA can first form RNA and then can form protein. Anyway, this is what when we discuss about DNA versus RNA. When we discuss DNA versus RNA, the most important concept. With the second concept, we are considering DNA as the best one. Because if a genetic material is unstable, if it is highly reactive, it creates a lot of problems with sudden changes and mutation. So we are considering DNA as the best genetic material. Okay, and you know the presence of thymine in DNA is also giving additional stability to DNA. Okay, and it is said that evolution says that the first formed life forms were with RNA as the genetic material, and gradually, since it is unstable molecule, reactive molecule, DNA evolved around RNA, and the life forms were with what? Became with DNA as the genetic material. So, DNA versus RNA, I think this is clear. You can just criticize this to DNA and RNA based on 1, 2, 3, 4 important points. A genetic material must be able to replicate, it must be stable, and it must provide some scope for evolution, and it must be able to express as characters. Okay? Then, if these things are clear, the remaining topic, Hershey and Chase experiment, we will discuss in the next class. Okay, thank you.